So on behalf of our respected Honorable Founder President, Sir, Dr. Ashok K. Chauhan, Sir, and all the Amity fraternity, come to Professor Leah Silva, Department of Food Science and Technology, Slovenia, to our esteemed organization. From the University of Ljubljana in the year 1995 and her PhD in 2001. She has supervised more than 60 students and published more than 30 research papers. She has given more than 80 presentations at various scientific conferences around the world. Her research area includes extraction techniques from medicinal plants, followed by chemical and biological characterization like anti-diabetic, antimicrobial, and neuroprotective activities which are present in these herbal extracts. Besides, she is the scientific editor of Sensors and Transducers and guest editor of a special issue of journal on antioxidants, dietary polyphenols, and neuroprotection. Professor Leah, your presence here today signifies a remarkable opportunity for our students and faculty to benefit from your wealth of knowledge and expertise. With these words, I would like to invite Professor Leah to kindly share her presentation on the topic, extraction and characterization of bioactivities from invasive knotweed species. Professor Leah, please. Thank you.
Hello? Okay, it all works well. First of all, thank you very much, Professor Charagupta, for this kind introduction and even before for the invitation. It's my first time in India and I'm appreciating it so far. I'm pretty sure that this is not my last time here. As Professor already introduced me, I'm coming from Slovenia. So can you tell how many of you ever heard of Slovenia before? Just raise your hands. Who heard of Slovenia before? No problem. I know that, okay? I know that, okay? It's not a problem. We are so small that you don't need to be ashamed of the fact that you don't know that. That's why I'm here, for you to know one more country from the world. It's a tiny country, so obviously it's in Europe, because this is Europe that I'm sure you all know. And it's this dark green spot. And we have, as you can see, only 2 million population, which is nothing for you, I know. But anyhow, any person is special. So we are also proud to be from Slovenia. So this is the shape of our country. It's surrounded maybe with some a little bit more known countries like Italy. This would be Italy, Austria, Hungary, and then Croatia. So for you, just to more or less, it's more or less Central Europe. We are members of European Union. And there is a funny thing. If you see this advertisement that we are having, I feel Slovenia. And if you check better, we have the love in the name of our country. And we are very happy of that. So if you ever think of Slovenia, think of love. There's just some photos from some of the places that we like to show to the people that never went to our country. And we hope that that's something to attract people to come, because it's a very beautiful country. With since we are so few, we have lots of nature. 60% of our country is covered with the woods. And on top, we have nice lakes, the caves, and we have a tiny piece of the sea, Adriatic Sea, where you can go and jump, to ski, to snow, to swim, and you also can make winter activities. At the moment, we have a winter with the snow, so you can do also winter activities there. this is not as high as Himalaya, of course, but anyhow, almost 3,000 meters high. So we have snow during the winter. And now I'm moving on to my university, University of Ljubljana. Ljubljana is a capital of Slovenia, and this university is our biggest university and the oldest. It made a little bit more than 100 anniversary. It was founded in 1919. And we have about 40,000 graduate and postgraduate students. Considering that we are only 2 million, this is a big number. So this is the biggest and the most, the best university of Slovenia. It's similar to MT, so we cover all the areas of science. So we have 23 faculties, 3 arts academies, and we are listed among the top 500 universities on Shanghai rankings. So we are also not too bad regarding not the teaching and not also about the research. And these are some numbers about the projects that I think you are not that interested. Okay, so I'm coming from biotechnical faculty. These are the photos. This one is to prove that we have snow. So also at our faculty in the winter, sometimes it's with the snow, not always, but sometimes. We have about 2,700 students and various studies, agronomy, biology, forestry, landscape architecture, wood technology, animal science, food science, and technology. That's where I'm coming from, and I think that's your field of study too. So that's why I'm giving the lecture to you. And then also biotechnology and microbiology. I'm teaching, as Professor Gupta already told, chemistry, because I'm a chemist. And I'm teaching not only the food technologies, but also biotechnologies, agronomy students, and so on. So I have a broader spectrum of the students, and they are actually the same as you are now. I'm really happy to see that students all over the world are really very similar. So that's very good also for me as a speaker. Okay, I'm here in the framework of some exchange program, which is called Erasmus. That's some European program that encourages international exchanges. And at the beginning, that was considered just for the members of European Union, but now they are trying to promote also the exchanges to outside Europe. And I came already in the framework of this Erasmus scholarship. And you as a student have the opportunity to also ask for the scholarships, for instance, to come to make a 1 semester or 2 semester at our faculty. Just if someone will be later interested, you can contact Professor Gupta and she will give you all the information regarding that. Yeah, I will not go more in detail in this. I'm coming from the chair. So this Department of Food Science and Technology has several chairs. One is biochemistry and food chemistry, which is where I'm seated. Then it's biotechnology, microbiology, food safety, meat technology, food assessment, plant food technologies, human nutrition, and enology. So I think you probably have a little bit similar of subjects in your study too. And now I'm moving to the subject of my research work. This work is made in a bigger portion with the help of the students like you are, or the bachelor's or the master students. They were making each of them one part during their thesis work. So first, not wheat. I don't know if anyone is familiar with this plant. I probably not. It gives lots of trouble. In Europe, in the States, it's listed as one of the most invasive species in the world. It was brought to Europe from Japan. and where it's not invasive, of course, it's native there, but it didn't have natural enemies in Europe. It started to spread too much, and it's killing the other native species. Okay, we have in Slovenia and also in Europe, this is not just Slovenia, but all around Europe, 3 taxons: Japanese, Sakhalin, those are originating from the east. And then we have still one called Bohemian knotwing, which is the cross, the hybrid of first 2. So it was only born in Europe. And that one is unfortunately the most invasive. So how does it look like? It's some kind of bush. It disappears during the winter with the cold temperature, and then it starts to regrow again. In the late summer, it gives the flowers. And these are some differences among the 3 species. We had help of some biologists to identify all 3, not to have a mess then with the results. Anyhow, this is what the trouble that this plant can make. It can penetrate all around, and it can really make a big damage. There are several. So it was introduced to Europe in 1850, as you can see, as an ornamental plant. But then it started, and it's really difficult to get rid of. You can try to use some herbicides, some steam sterilization, or some introducing some insects or some fungi. But unfortunately, the success is limited. But sometimes if you have some trouble, you have to search if you can use the trouble in your profit. So why can this plant be good? It was used traditional in Japanese, in Chinese medicine as a wild edible vegetable. So, and that already gives us some indication that the plant might have some activities that we could use in the future also, or as a medication, or as food supplement, or anything like that. And it was found that it's a good resource of one polyphenol called resveratrol that I'm going to talk about later much more. So maybe just this was skipped. So resveratrol is some polyphenol. I think you already heard of polyphenols. They are important molecules. And resveratrol is something that is contained in the red wine, for instance. That's for at least in Europe, the most known source of resveratrol is the wine, but there are some other sources. Okay, the grapes that gives the red wine, of course, the peanuts, blueberries, dark chocolate, and so-called itadori tea that is prepared out of the knotweed. And you can also go to the shops to buy some food supplements. And if you go to search for the food supplement for resveratrol, if you check better, the source is usually already knotweed. So they're already extracting resveratrol from this plant. Okay. Okay, now just this is too small, but you know all these things mostly already. So this is about polyphenols. We know that they have several different activities, and this has been widely researched. It can have different activities: anti-diabetic, anti-inflammatory, anti-neuroprotection, or something like that. And that gives us idea to also check the extracts of these invasive plants. So extraction, you know how it works. So you have some plant material, you add the solvent, you mix. Usually what you do is first to put to mill, to dry, freeze dry the tissues, to release then the substances that you want to extract. And then, of course, you have to add the suitable solvent. And the solvent that you add has to be regarding the polarity similar to the substance you want to extract, right? That's the basic principle of extraction. So if you want to extract the polar molecules, you would add the polar solvent. If you want to extract the non-polar molecules, you will put the solvent that is non-polar. But mostly, as in the case of polyphenols, the molecules are something in between. So you add the solvent that is in between. So let's see what we did. Okay, this one is too fast for me, it seems. Okay, we made an optimization of extraction, and it turned out that 50% ethanol and 50% acidified water was the best extraction media. So ethanol is less polar than the water, and acidified, we added 2% acetic acid. It's because there are reports telling that the polyphenols are more stable in the low pH. So what you want to do is to obtain as much as those substances as possible, right? Because if you add the wrong solvent, the most will be trashed. Okay, and what you want is to have the solution that contains the maximum amount of the substances that you want to then profit from. Okay, we perform a two-phase extraction, which means that you add the solvent, mix in some way. We accelerated the extraction with the ultrasound, which usually reduces the time of extraction. It's widely used. It's a simple technique, and it's widely used for that. And then you separate the extract from the non-soluble matter, and you add another portion of the solvent to the same undissolved matter, because that is more efficient than if you add the full amount of solvent at once. And then we were making different analysis of, I just named here some, but I will, during the lecture, give you results also of some others. So antioxidant activity, anti-diabetic activity, neuroprotection, neurotoxicity, and we also performed the chemical analysis. So we identified and quantified several polyphenols that might attribute to these different activities. Okay, first, chemical characterization. The first round, we just chose 4 polyphenols that were the most easy to detect, and we knew that they are present in these extracts. Resveratrol that I already mentioned, then it's polydatin, catechin, epicatechin, and if you check these chemical formulas, you can already see that they are, I already mentioned, polar, non-polar. These substances are somewhere in between. So non-polar part is these rings, and then the polar parts are OH groups. And then, I don't know this image is quite small, but if you check resveratrol and polydatin, can you see some similarities of these 2 molecules? Anyone? I don't know if you can see. Are they similar or not? In some part, is one part very similar. But this part, right? So this is the same as this. And what is this? Do you know what is this part? What would this be? How does it look like? Like sugar. Perfect. That's it. Exactly. And this is very often how you will find the polyphenols in so-called glycosylated form. So it's not just polyphenol, but it has also some monosaccharide attached. And actually that form is more stable. So if you find that form, it's very okay, because that molecule is then more stable than, for instance, in this case, resveratrol itself. And now let's see the results. So this first is epicatechin and catechin. it's in Japanese knotweed, in Bohemian knotweed. Okay, they are presented in both. This is the roots of the these two knotweeds, and then it's polydatin and trans-resveratrol. So the one that is present in the highest quantity is not resveratrol, right? So if we would be checking just resveratrol, which is this one, the smallest, we would say, everybody is wrong. There is no resveratrol present in the tissue, but it's not true, okay? It is present and in high quantities, but in a glycosylated form. So sometimes you have to be careful. What are you checking? Because as you know, this was performed by HPLC analysis. And with HPLC analysis, you always can detect only what you are searching for, particularly if you have, this was just with a simple DAD detector. You know what is diode array detector? It checks the spectrum, yes, absorbance spectrum. So if you have the detector like that, you don't really find new substances. You are just finding the ones that you are searching for. So, and we were just, in this case, searching for those four. We found them, and we found the high values, and particularly the one polydatin, which is very good. And this, one more thing, it's important to express the results to the dry weight. I know that in food technology, sometimes the results are expressed to the wet weight, but you have to be careful because during processing or during maturation, sometimes the quantity of water changes in the tissues. And sometimes we tell that something raised, but it didn't raise actually, if you divide that by the dry weight. So that's just one comment maybe also to mention. Okay, let's go forward. So this study was then performed further on by not with the help of the institute that is specialized for the chemical analysis. So it was also HPLC separation, but it was followed by mass spectrometric detection. And together with the two students were working that making their master's degrees with, and these are the results. So they managed to identify lots of more polyphenols. Not only identify, we also already have the results of the quantifications in all three taxons and also in different tissues. So in the roots, that's what's the most interesting, in the roots, and also on the young offspring. So as soon as the plant starts to grow, that's a very good source also of these chemicals. So, and as you can see, if you were already listening about polyphenols, we have different groups of them, and we found polyphenols of really very different groups. I will not go in further detail of the particular quantities of each of them, because that would take a full lecture, but just for you to have an idea how the study grows. Okay, you first start with something small and then it grows to something much bigger. But of course, there is lots of work, in this case, from the students and from the mentors and all that. Okay, let's now go to the bioactivities. First of all, antioxidant activity. That's one activity that with the polyphenols, it's something that you have to do initially, right? But there is some trouble because there are lots of methods for determining antioxidant activity. And if you make 5 different methods, each method will give you different results. So now what? Okay, there is one important thing. It's always better to perform more than one. Why? Because they follow different mechanisms. Those methods, DPPH, ABTS, and so on, there are really lots of. They follow different mechanisms. So if one antioxidant gives a good result with one method, it doesn't mean that it will also give it with the other. So it's always better to perform more. Okay, the first one was performed with this DPPH test. And as you can see, this is Japanese, Sakhalin, and Bohemian. And it's the rhizome and the young shots. And as we can see, basically the rhizome was always better source of antioxidants. And these are result of so-called cellular antioxidant capacity assay. And that one was performed on the yeast. So the yeast was treated with these polyphenols, and then you check if these polyphenols are able to have the antioxidant capacity inside of the cells. And in this case, as you can see, this is Bohemian and Japanese, and the rhizome of Bohemian and Japanese. And Bohemian was more potent than the Japanese not wet, which is in the contrary of the results of here. Okay, anyhow, but why are these experiments also important, besides of the ones that are pure chemical? Because this is pure chemistry. The first test is pure chemistry, is the interaction between antioxidant and the radical. The second one also takes into account the penetration through the cell, through the membrane of the cell. Okay, and that's important because what we want is to have activity inside our body. Okay, so it's not important to have the activity just inside the sample. We also have to check if this would penetrate our organism or not. We call this bioavailability. And this is one way to check if antioxidant has also the activity inside the cells. So this is the complementary result. It's not that one is replacing the other. You have to make both. Okay. Okay, the next one, anti-diabetic activity. So you already heard about alpha amylase, right? That is an enzyme. Where do we have that enzyme? Where is this enzyme present? Do we have it? Yes. Where? In the mouth, in saliva, exactly. And it degrades what? Carbohydrates, starch, for instance, right? Exactly. And people with, and the final product is glucose. Perfect. Very good. I make a question to my students and then I'm checking in, and you did a good job. Very good. Okay. So the glucose is the one that gives trouble to diabetic patients, right? And particularly the sudden changes. If the amount of glucose changes suddenly, then they might be in trouble. And there are already medications that they can consume together with the food that would inhibit a little bit alpha amylase. It would reduce the activity of alpha amylase. But of course, we are trying to find the natural sources to do the same thing. So we tried also with the extracts of knotweed. And I'm happy to present you the results because we were successful. We can see that these are all three different exons with three different concentrations, and this is the inhibition of alpha amylase. And we can see that all three were inhibiting alpha amylase, all three extracts. And Japanese and Sakhalin were a little bit better, particularly in the lower concentrations. But already at the concentrations of 1 milligram per milliliter, they were inhibiting almost, particularly Japanese and Sakhalin, not with extracts, were already almost completely inhibited alpha amylase. So it can be used, obviously these extras could be used to help diabetic patients not to have those sudden changes of glucose in their blood. Okay, one of the students, this was not done on our faculty, one of our students went to exchange with also with this Erasmus program to Prague, which you may have heard of already. Prague is a city it's not too close, but compared to distance to here, it's close to me. It's in Czech Republic. And they had the laboratory when they could grow the cancer cells. So we wanted to check if our extracts would also have some anti-cancer activity. So what we were checking, the rhizome extract, in this case, we were checking the flower extracts, and the first results are non-cancer cells. So, and these numbers represents the concentration that was high enough or low enough to kill half of the cells. Okay. And all these results represent results for the different cancer cells. Okay. So now focus, please, first. So these results are from the healthy cells and results from the cancer cells. And the values for the normal cells were lower. So we need less of extract to kill the healthy cells. Then we need the extract to kill the cancer cells. So were we successful? What do you think? Is this okay? So you would put 30 milligrams per milliliter and half of the healthy cells would die. Okay? But half of the cancer cells will die only if those values would be higher. So is that okay? It's not okay. Okay, you first kill your good cells and only after in higher concentration you will kill the cancer cells. You don't want to do that, of course. What you want to do that is to kill the cancer cells at lower concentrations. Okay, you have to make an experiment to find things, right? Sometimes your results will not be as you want. But don't worry, it's also the result. This was published in the international journal. Okay, that's important. I think that's also one important information that I want to share with you with this lecture. Okay, anyhow, but it doesn't matter. One extra doesn't need to be good for everything, right? We are not expecting that. But we also checked antimicrobial activity. You surely also have microbiology, right? You are better at microbiology than me, surely, because I'm not a microbiologist, but I have, again, the colleagues that are on our department, and there was another thesis that was made by our student, and she was checking so-called minimal inhibitory concentrations or minimal bactericidal concentrations. And as you can see, we were against 3 different bacteria, gram-negative, gram-positive, and the fungi. And as you can see it with all 3 different taxons, and they were all able or to kill or to inhibit the growth of bacteria, which is very good. So our extracts have antimicrobial activity. So this was a very good result, in the contrary of the previous one. And as I said, this was published in the international journal. I will share this presentation with you through the Professor Gupta. So if someone is interested, you can read these things more in detail. And I will move to the last part of my talk. I still have some minutes. Okay, so neurodegenerative diseases, it's Parkinson, Alzheimer's, and many others. And the problem of these diseases is that they occur during our lifetime. And when we have the symptoms, we can't treat them yet, not that much. There is some medication now already going around. Let's see if it helps. You can eventually reduce the speed. But what we want is to prevent. And the idea is to prevent it by eating healthy. And actually, I think your diet is healthy regarding the consumption of antioxidants because it's plant-based diet, okay? So regarding that, and that's what these are just some facts that I will not even go through. I will just go to the results. And this was performed in the laboratory of Professor Silva that is also present here. He will not give you a lecture today, but he will give it tomorrow in another camp, in another faculty. Yes. Anyhow, I will just go fast. So we were growing neurons, the cells, and then we were treating them with the not with extracts. And we were checking after this was still the initial experiment. So after 48 hours, we were checking how many cells died out of necrosis or apoptosis, okay, to see if these extras would be neurotoxic. So if you treat the cells and if they die, the extracts are toxic and we don't want it, right? And the other thing is to check neuroprotection, if they can protect the neurons from the cell death caused by the oxidative stress. So we were treating again, but not with extracts 24 hours. Then we were adding reactive oxygen species after 24 hours. And we were checking again if these values increased or decreased. And let's see the results. So this would be apoptosis, necrosis, and these left columns are for the neurotoxicity. This is the control, not treated. This is Bohemian, Sakhalin, and Bohemian, Notwin. And we see that the apoptosis increased a little bit by treating the cells with the extracts. But at the same time, necrosis like that decreased. So in overall, not more cells died. So we can tell that the extracts are not neurotoxic. That was one conclusion. But then let's focus to the right columns. So to these ones that are now indicators. This would be control. Of course, more cells died when we were treating them with reactive oxygen species. But when they were treated previously with the extracts, the percentage of apoptotic cells was much lower, as you can see. And similarly, also the necrosis-like cells. So was this result good? What do you think regarding neuroprotection? Yes, this is a very good result. So our extracts were able to reduce the cell death caused by oxidative stress. Why is oxidative stress is connected with neurodegenerative diseases? Because that can be one cause for the neurodegenerative diseases, okay? And here is just the summary of all that I told already, but since I'm running out of time, I will just thank you for your kind attention, and once more for opportunity to visit your very nice university. And I'm almost sure that I will come again. And I will be really happy if anyone would also come to visit our university. I will share the classes that you can attend at our university. I think they have to be quite similar regarding the subjects as yours, and you get some new experience that might make a big difference for your future. So thank you very much. Yes. Yeah. Or not?
Yes. Yeah, thank you, Professor Silva. And now for this insightful presentation, I hope students must have gained knowledge about how to carry out the extraction and to carry out the biological activities. What are the different aspects of the activities that have been looked upon? So now you can ask the questions. Students are free. Anybody, if you have any query related to the topic, or if you have any query related to your her university, like Slovenia, she told you that there are various courses running, particularly in food science and technology. So for your high research studies, if you are interested, any query regarding that, you can ask.
You can also reach me through email. That's not a problem. It will be shared by Professor Gupta. So it's at any time, if you will have a question, you can always reach me. So it's not a problem. And I think, okay, I think there is still one slide with more publication. You will share. No, you don't need to do anything.
Okay.
It's the last slide is some more publications that we published. And if someone is interested, please welcome to read.
So I'll share her slides, whatever the presentation she has given to Dr. Monica, and you can just, if anybody student is interested in this research, so you can just take from her.
Yeah.
Okay, so no questions? Yeah, ma'am, please.
No, I like that.
Okay.
Like, it's a bead actually.
Yes.
Bead.
Yes. Yes.
So are these toxic studies done on this bead? Because if we will be using it in the formulation of like
Yes.
nutraceutical or any functional food.
Yes.
So I think we have to do the toxic studies also.
Yeah. If the plant was already used traditionally, then you don't need to perform the toxicity studies. And this plant was used in traditional, in Japan and in China, it's used as an edible plant. So then you don't need to perform the toxicity studies,
Okay.
at least by our law.
Yeah. So I think it's same in India as well. Actually, some traditional plants are there,
Yes.
some are underutilized or some weeds are there.
But anyhow, it's always nice to make. So if you are interested in making those studies, it's always nice to make. It doesn't harm to make
Okay.
any
Yeah.
more studies.
More
It's like
studies,
that.
yes. And my second question is, have you developed any functional food out of this or adding some extracts and all? Because I think this plant is having an excellent nutraceutical properties, antimicrobial and other
Yes.
anti-cancerous properties as
That
well.
is the next step anyhow, but we were already, some students were making some muffins out of these extracts, and then they were checking also by sensory analysis, because that's also important. It's okay, one thing is to consume it as food supplement, and the other is to introduce it with the food.
samples. Yes.
Exactly. And then on top of the activity, you have also to check the sensory.
Yeah, sensory.
Yes.
That is more important actually.
Exactly.
Yes.
So yes, there was, but it's not still in the form to be shown. But anyhow, we were doing already.
Definitely.
Yes.
So we'll be in touch. I think you're doing an excellent work on different plant
Thank
species.
you.
So we'll be in
Thank
touch.
you.
Thank you. Thank you.
Thank you.
What are the different programs that you're running for food science and technology?
The study programs.
Yeah.
You mean, we have the bachelor's, so bachelor's and the master program. So it's 3 years bachelor's and then 2 more years to reach the master's. And it's the first, the bachelor's is the combination of food technology and nutrition. Then on the master level, the study splits. So we run separately the food technology and the nutrition. We have more or less, on the first year, we have about 70 students. Then on the second, I think we have 60 on each program, something like that, 60 food technology and 60 of nutrition, something like that.
So we'll be in touch with Dr. Gupta. I think
Yes.
you can apply for that Erasmus and all.
Yes.
That kind of program we can have with your university.
Yes.
Okay.
Definitely.
Yes, of course. Thank you.
Thank you. Thank you.
Thank you, Professor. So now on behalf of Amity Fraternity and Amity Institute of Food Technology, we'd like to present you a small memento, Amity.
Okay, perfect.
Can you make some more notes? Yes,
Yeah. Yes, but also the students and everything. It's possible. Just
you can also come down. Please join us. Please join us. Thank you. Thank you. Thank you. You can
So good afternoon, students. On behalf of our respected Honorable Founder President, Dr. Ashok K. Chauhan, and all the Amity fraternity, I extend heartfelt gratitude to Professor Leah for gracing us with her insightful lecture. Your expertise and global perspective have enriched our understanding and broadened our horizons. We are immensely grateful for your time, knowledge, and dedication in sharing the valuable insights. And Madam, I would really like to tell you it was a very, very interesting, presentation, and you have explained the various concepts so beautifully that anyone, even the undergraduate students can, understand the concept. So really thankful to you for that. Your presence has not only inspired our students, but also strengthened our commitment to fostering international collaboration and learning. We look forward to future collaborative opportunities to engage with you further. Thank you again for your enlightening contribution. Thank you very much. And we look forward and we thank Dr. Charu Gupta and our own faculty also, Dr. Monica, for organizing this lecture. Thank you. Thank you so much.
close the meeting.